Thanks for the kind introduction and for the invitation to speak here. And so I want you to take a moment just to think about what you saw in the morning when you looked in the mirror as you examined yourself and thought about perhaps what it is that makes us human. When I did this morning, I saw an organism that's just 43% human, and not just because it was early and I hadn't had my damn coffee yet, but when we think about what makes up our human bodies, each of us has about 30 trillion human cells, but we have about 39 trillion microbial cells, and that's where that 43% number comes from. But you might think, well, wait a minute, it's the 21st century, who really cares about counting cells anymore? Let's think about it at the DNA level. And when we think about that, it's even more astonishing, because to our 20,000 human genes, amazingly, the size of our microbial gene catalog ranges from 2 to 20 million microbial genes. And so by that measure, we're at best 1% human, and we're neglecting 99% of the system when we ignore this microbial side of ourselves, which contains most of the unique antigens and most of the unique biochemical capabilities. So this really makes us re-examine the concept of humanity. And for example, what if I told you there was a drug that could change most of the genes in your body? That would be pretty remarkable, right? But it turns out to be literally true when we think about those microbial genes. And work from Pierre Bock and his collaborators at EMBL has shown that a huge number of drugs, not just what you think of as antibiotics, have radical effects on organisms within your microbiome, changing their species abundance and even changing their gene abundance. And so this idea that our microbial genes are genes that we can reshape during our lifetimes rather than ones that are fixed at conception, like our human genes, is a breakthrough that's going to be incredibly important for individualized medicine. So the gut microbiome is not only affected by medications we take, but it also affects our response to those medications tremendously. And some of the stories that have been well worked out include our response to liver toxicity for acetaminophen, our response to digoxin, whether or not we have particular strains of Eggotella lenta that degrade it, anti-cancer drugs like cyclophosphamide, cisplatin, and even the latest PD-1 and PD-L1 checkpoint inhibitors. Whether they will work for you and whether they will be safe or effective depends tremendously on the microbes that you have in your gut. And what's more, in 3 papers that came out in the same issue of Science early last year, not only does the microbiome explain whether those drugs will be effective for you, but you can transplant those characteristics from humans all the way across the species barrier into mice by transplanting the microbiome, and then start to do precision medicine by asking what treatments work on those mice and doing the translation back into humans. And there's a tremendous amount of excitement about that here at UC San Diego and at the Moores Cancer Center for a number of different indications. So how does the microbiome do all this stuff? Well, the vast majority of your microbes are in the gut, but it's important to remember that the gut is not Vegas, and that what happens in the gut does not stay in the gut. And the effect of those gut microbes pervades the entire body, whether you're talking about things like IBS and IBD, which are maybe obviously linked to the gut microbiology, to things that are less obviously linked, like food allergies, like liver disease, like cardiovascular disease, where Stan Hazen at the Cleveland Clinic and his collaborators have worked out a very nice story surrounding dietary sources of choline and carnitine that are metabolized to trimethylamine by gut microbes and then converted to trimethylamine N oxide in the liver, which then contributes to cardiovascular disease. And perhaps most excitingly, we're starting to see connections between the gut microbiome and the brain, with the gut microbiome affecting immune signaling, affecting chemical signaling, affecting epigenetics, for example, via butyrate, which is a potent HDAC inhibitor, but also by other pathways. And then finally, by a direct neural connection via the vagus nerve, which can react to gut microbial changes on very short timescales. So a really important question is, what is our stewardship of all of those microbes in the gut? And when we study ancient populations like the Hadza hunter-gatherers, which we've studied with Jeff Leach and Justin Sonnenberg and others, what we find is that the Hadza and other populations living what we'd call traditional lifestyles have totally different gut microbiomes from anyone who's taken up farming, let alone anyone who's living an industrialized lifestyle. And if you look at the scale from least integrated into a market economy to most integrated, what you see is entire phyla, like the spirochetes that were once prevalent, but that we've lost completely from our gut microbiomes in industrialized societies. And this raises a really important question, right? Are we taking this beautiful rainforest that's our co-evolved gut microbiome that we've spent millions of years evolving, and have we just destroyed it, turning it into like an urban landscape where only the rats and the pigeons survive? And it is in fact true that our gut microbiomes are dramatically less diverse than those of the Hadza, those of the Yanomami, those of the Inuit, and other peoples living traditional lifestyles. So this raises a really important prospect that we may be facing a global microbial biodiversity crisis. And I'm sure you're all familiar with Rachel Carson's work documenting the problems that DDT caused in the large-scale physical environment around us. And more recently, Marty Blazer at NYU has sounded a similar warning call surrounding our inner ecosystems in his book Missing Microbes, which I definitely recommend you read. And what's fascinating is that as infectious diseases were brought under control during the 20th century through advances in public health and medicine and so forth, at the same time we see this dramatic rise in what are usually considered non-communicable diseases, so type 1 diabetes, asthma, multiple sclerosis, Crohn's disease, and many others. And what amazes me the most is that in 2002, when that graph was published in the New England Journal of Medicine, none of those 4 diseases were known to be linked to the microbiome. Whereas today we know that all 4 of them and dozens of others are not only associated with the gut microbiome in humans, but can be caused or cured by manipulating the gut microbiome in animal models, especially with transfer of human microbes into germ-free mice. So to start to get to the bottom of this and to characterize what a healthy human microbiome looks like, in the Human Microbiome Project, together with about 400 researchers around the country, so this was a very large-scale project that many different people played important roles in. We characterized as a starting point about 250 people at up to 18 sites in the body and collected 4.5 trillion bases of microbiome DNA. So if you think about that, that's 1,500 human genome equivalents, which was a tremendous amount of data for 2012, which is why it costs so much. And so the great thing about this project is we had an unprecedented amount of DNA sequence data concerning the human microbiome. But that was also the terrible thing about the project. And to illustrate this, here's the first file of data from the HMP. There's another 17,000 files like this one, and this is the first 0.1% of it. And despite the fact that what we're doing is fundamentally ecology, I bet you can't tell who lives where in the ecosystem from that, right? You probably can't even tell it's an oral sample, let alone the sequence signatures that let us determine that. And this is a huge problem clinically now that various companies and also citizen science projects like the American Gut Project that I run here at UC San Diego offer individuals a way to get their microbiome sequenced. And from a clinical perspective, for those of you who are clinicians, I'm sure it's your nightmare that your patient is going to come in with a huge grin on his face and this list of 1,000 species that they found in his gut with microbiome sequencing, and expect you to tell them something useful about that in the 12 beautiful minutes you have together, or whatever it is now. And I mean, what are you going to do for that patient? Maybe refer them to colleagues in psychiatry for being so crazy that they thought you could do something with their list, right? And so the challenge we face is to make it not crazy and to give you something you can integrate in a few minutes and make some sort of decision based on. So using tools we developed in my lab that I won't get into here, but notably the CHIME pipeline and the Cheetah database that links it all together, we took all that data from the Human Microbiome Project and turned it into this map. And what this is a principal coordinates reduction using a distance metric called UniFrac, where we measure the distance between communities based on evolutionary history. So basically, each point on this map is all the complexity of a microbiome read out from its bacterial DNA and distilled just down to 1 point. And 2 points are close together if they're more similar in terms of the evolutionary history of the microbes they contain, and they're further apart if they're more dissimilar in that evolutionary history. So these were all healthy subjects, so there's no disease on this map, but you might wonder what makes a difference to the microbiomes of these 250 healthy people. And if I show you the main factor on this map is the color, it's immediately obvious what's going on, which is that the mouth, the skin, the vaginal, the fecal communities are very different from one another from a microbial standpoint. And if I highlight the microbes of the first person in the HMP, you see their mouth and their gut are in completely different regions, like different continents. But it wasn't until we did the Earth Microbiome project, where we crowdsourced tens of thousands of samples from the scientific community around the world, that we really understood how profound these differences on one person's body could be in terms of the microbial ecology. Because what we could do is we could go out on the planet's surface and ask what 2 points on the planet are just as different from each other as the mouth and the gut of this one individual in the HMP. So if you think of your mouth maybe as being kind of like a coral reef with these complex mineralized structures covered with biofilms that perhaps your dentist pesters you about, the amazing fact is your mouth is as far from your gut in its microbiome ecology as the water in that reef is from the dirt in this prairie. And that's amazing, right? Because less than a meter along your body makes as much difference to your microbiome ecology as thousands of kilometers across the Earth's surface, despite the fact that they're two ends of the same tube. And we never expected that. But the important thing here is you have to think not just of your patient having one microbiome, but having a whole archipelago of different microbiomes. And we're just starting to understand how they communicate. So in the American Gut Project, so the great thing about the HMP is it really provided this baseline for what healthy microbiomes looked like. But the exclusion criteria were very strict, and you could only participate if you happened to be on site at WashU or Baylor. And what we wanted to do was to open microbiome science up so that anyone could participate. So in 2012, we launched the American Gut Project using crowdsourcing and crowdfunding so that each subject could add themselves to the research project by supporting the cost of adding their own samples so we could scale it indefinitely. Of course, it turns out that not everyone wants to know what's in there. So these are middle schoolers touring our lab, learning we're going to use lasers and robots to read out the DNA of the bacteria in their poop, all of which is 100% accurate. You can see a certain amount of response heterogeneity there. We don't yet know if that's linked to the microbiome, but sooner or later we'll have the sample size to find out. But some of the things we're finding in American Gut. So the idea that you are what you eat is certainly not a surprising idea, right? So this painting dates back to the 1500s. But what we were able to see when we quantified that in more detail is that the effect size, so these are power curves where you have the number of people per group on the x-axis and the statistical power of the test on the y-axis. So this is 80% power to detect, this is 100% power. And so with a few dozen people per group, you can see things like age, inflammatory bowel disease, whether they used antibiotics. And then with hundreds of people, you can get things like how much you sleep, which you might not have known the microbiome was involved in, but it is. How fat you are, how much you drink, how much you exercise, or I should say how much you say you exercise, because all this is self-reported data, right? So either there's an effect of exercise or an effect of lying about it. But the steepest curve on this graph is the number of different species of plants that the subjects ate in the couple of weeks before they sent in their sample. And that has a much stronger impact than a lot of the other than any other variable on this plot. And what's amazing about that is that the number of plants that you eat, number of distinct kinds of plants, has a much bigger effect than whether you self-report as a vegan or whether you have something like diabetes or Crohn's disease or other diseases that we can pick up, as well as most of the effects of most of those medications that you saw that you saw earlier on the slides at the beginning. So what's amazing about this is lifestyle factors that you have control over, like the diversity of plants in your diet, can have a large impact on your microbiome, and now we're trying to connect that to health. So the most compelling case we have about why you care where you are on the sort of microbiome map is in fact an infectious disease example, and it's Clostridium difficile associated disease. And this is work we did with Alex Karutz and Mike Sadowski at the University of Minnesota. And when we put this disease on the map, these spikes are fecal samples, but they look nothing like a healthy fecal sample, which is given by the HMP distribution at the bottom in brown. So these are patients with C. diff, and you can see that their fecal gut microbiome looks absolutely nothing like a healthy gut microbiome. And what's going to happen is that 4 of them are going to get what's called a fecal transplant from one donor, who you can see is down in the healthy distribution. I should note that these were done with different sequencing technologies and different methods, but we were able to develop techniques to integrate the data into one view of these different cohorts. So anyway, 4 of these patients are going to get a fecal transplant from this one donor. If you've been living in a cave and you don't know what a fecal transplant is, here's Bill Sanborn, our chair of GI, very proudly about to deliver his first one using hospital-grade stool that he got from a nonprofit called Open Biome. Because remember that the FDA regulates fecal transplant as a drug. It's difficult to prove that you manufactured it under IND, right? Under GMP rather, when it's literally something your subject pooped out. But you have to show you put in an effort. Okay. So anyway, so 4 of these patients are going to get a fecal transplant from that one donor. And in this animation, each frame in this animation is 1 day in the gut microbiomes of these patients. And you might even wonder, are we going to see anything by adding a few grams of stool to their microbiome ecology, which is already so complex, even if it's dysfunctional? And so then the question is, how completely do they resemble the healthy stool? And then is it a fast or a slow progression? How complete is it? Do they go straight there, or does something happen along the way? And what you see is that immediately, so in just 2 or 3 days, their entire microbiome ecology resets to the healthy state. This is coupled to remission of all their clinical symptoms, despite the fact that they'd failed antibiotic therapy for C. diff for 2 years to be eligible for this trial. And you can see that they stay in the healthy microbiome ecology state for months of follow-up. But what's more important than this specific example is the general concept that it provides, that we can take all that data, all those billions of sequences and those thousands of species, and turn it into a graphical display that's very understandable, where basically you identify a problem with the microbiome, and you can fit one axis from sick to healthy, and you can track people's progress towards healthy in a way that you could imagine understanding yourself and that you could imagine explaining to a patient. And so the challenge that faces us now is for all of these other different diseases that have been linked to the microbiome, some of which have obvious effects on the microbiome and some of which have subtle effects. How can we in the same way identify what the dysbiosis is and then track its progression with treatment into the healthy state? So the first thing to do is to find the good and the bad places on the map, which as a community we're now pretty good at doing, and we've reached out through the Center for Microbiome Innovation Seed Grant program to medical specialties across campus to be able to get a lot of samples from a lot of different indications. But we want to go beyond mapping. We want to develop a kind of microbiome GPS that tells you not just where am I right now, but where do I want to be on the map, and what do I want to do turn by turn in order to get there. Whether it's something as radical as FMT or phage therapy, which we're also doing at UC San Diego, or whether it's something that seems as gentle as diet, which can completely reshape your microbiome, but over a span of months, not over a span of days, or whether it's something in between, like improved drugs or probiotics or prebiotics, or all of the other things in the toolbox. So we want to develop this kind of microbiome GPS, and then because my main appointment's in pediatrics, ideally we want to make it so easy to use that even children can use it. So you could imagine a smart toilet where as soon as you flush, it delivers your latest data dump, as it were, to your smartphone, which, let's face it, I bet you're using in there anyway. And then just tells you where are you on the map and where did you go since the last time you entered data, as it were, and is that taking you to a good place or a bad place? And we took this photo when my daughter was 2, at which point she'd already figured out not just how to use a smartphone, but where we all too frequently use them. At that time, we couldn't do this kind of thing on a mobile device, but now that she's 7, we can do this in a web browser on a mobile device. And this is her own microbiome, developing from the day she was born over the first couple of years of life towards the healthy adult state. And again, this is the kind of thing that you can explain to a 7-year-old. You can almost certainly explain it to your colleagues or to a patient. So our ambitions stretch even beyond this, though, and our dream is that as you examine yourself in the mirror in the morning, that this is no longer going to be the realm of million-dollar instruments, but something that's in a clinic or even in your home. So the concept is that as you breathe on that mirror, maybe your breath is going to be instantly chemically analyzed, say by mass spectrometry or NMR or by Raman spectroscopy. Obviously, we're still working on the user interface for the display of this part. And then translated using some of the deep learning technology that backs Google Translate into a microbiome profile like you'd get from American Gut, and then places you on a map like the map defined by the HMP and other large cohort studies, and ideally identifying any problems that you may be at risk for, and then either telling you to consult your physician or telling you what you might be able to do in order to avert the need to do that before it becomes too severe. And so then when we're dreaming about this, you can imagine that maybe your smart mirror is also going to communicate with your smartphone so that when you're faced with 1,000 yogurts in the supermarket, you can use augmented reality to zoom in on the particular product you're looking for and then grab the barcode and scan it to prove that you got it. And then you could also imagine the mirror communicating back, the smartphone communicating back to your mirror and reporting what you ate, where you were, who you associated with, how many minutes you spent outdoors, all these things that affect your microbiome, so that the mirror can show you visions of yourself 5, 10, 20 years in the future if you behave every day as you did today, or what you might look like if you did a little better or a little worse. So that probably sounds crazy, but most of the individual pieces of technology exist already, including things like the live video manipulation that can not just make you thinner or fatter, but also happier or sadder. And so bringing them together is a lot of what we're doing at the Center for Microbiome Innovation that I direct with a growing list of corporate partners, and also now over 130 faculty in their labs at UC San Diego. We're tackling difficult problems like these and trying to build the future, not just of microbiome research, but of microbiome applications. And a lot of people assume that this is a joke, but Peter Dorrestein and Alan Jarmusch actually built a prototype of that mirror. So what you can see is there it is, backed by a mass spectrometer, where as you blow on it, you're getting a real-time readout of the chemicals in the breath. So although it's pretty clunky, and right now the cost is in the millions of dollars, in the same way, the chip that's in your phone that you take photos with used to be at the heart of a multi-billion-dollar spy satellite, and a lot of what separates that is the ability to produce them at scale and the market that promoted that production. So we think that this is stuff that's science fiction now, but will be science fact before too long. So with that, I'd like to thank literally over 1,000 collaborators who've contributed to the various projects I showed you very briefly. So I'm sorry I didn't mention them all, or if you're in the audience that I may not have mentioned you specifically. The many amazing people in my lab who it's a true privilege to work with, our various sources of funding, and I think we're saving questions for the panel discussion. But thanks again for the opportunity to be here.